biología o la filosofía, suscita grandes debates, puesto que los científicos aún no se han puesto de acuerdo en dar una definición tajante de lo que es un ser vivo. Y hay muchos, porque la Tierra acoge a millones y millones de especies vegetales y animales, una complejísima biodiversidad, resultado de miles de millones de años de evolución. Pero no sabemos cómo comenzó el proceso de evolución, ni siquiera si lo que llamamos vida es una rareza estadística o, por el contrario, un resultado inevitable siempre que se dan las condiciones físico-químicas adecuadas. Tampoco sabemos cómo se produce la transición de un mundo inerte a uno vivo o si la vida se puede replicar en un laboratorio. Pero de todo eso hablaremos en este ciclo, en estas 2 sesiones que vamos a tener hoy y mañana, en las que se nos ofrecerán las claves sobre la búsqueda de vida en el espacio, los procesos físicos y químicos que llevaron a la aparición de la vida en la Tierra y el debate sobre lo que entendemos por vida. La mesa redonda de mañana también empezará a las 7, como hoy, y se titula Diálogos, ¿qué es la vida y cómo surge? Diálogos desde la filosofía y la ciencia, y estará moderada por la principal impulsora y coordinadora de este ciclo, Valentina Rodríguez Castro, encargada de comunicación del Donostia International Physics Center, que presentará a sus invitados, Izaskun Jiménez Serra, doctora en astrofísica, Juan Manuel García Ruiz, uno de los mayores expertos en cristalografía, y Kepa Ruiz Mirazo, que también está hoy con nuestro invitado. Kepa Ruiz Mirazo es investigador del Instituto Biofísica, profesor de Filosofía de la Ciencia y Filosofía de la Complejidad de la Universidad del País Vasco y reconocido investigador del problema del origen de la vida. Con él iniciamos hoy la charla junto a nuestro invitado de honor, Szostak, Premio Nobel de Fisiología o Medicina en 2009 y cuyas investigaciones actuales están centradas en desentrañar el origen de la vida. Con ellos les dejo y ya saben que después de su intervención podrán ustedes hacer preguntas o comentarios al respecto. Y sin más, les dejo con ellos.
Hear me? Yeah. Okay. So this is going to be in English, as Marilis said. The first session of a cycle that we're going to have today and tomorrow. And for me, it's an absolute pleasure to have here Jack Szostak. A privilege for me, and I think for all of you who gather here today. It's Jack Szostak's lecture that we want to listen to. So I'm going to be very brief in the presentation. Maybe I will engage in a discussion later on, and I prefer to spend time with the discussion afterwards than in your presentation, because if I start with the CV of Professor Szostak, we make use of all the time, and that's not the purpose of it. So I will just say 4 important things that I don't want to forget. So Jack Szostak is professor of chemistry at the University of Chicago and investigator of the Howard Hughes Medical Institute. This is his current position. His early research on telomere structure and function was recognized with the Nobel Prize, and also previously with the Albert Lesker Basic Medical Research Award. Later on, in the 90s, he developed with other colleagues in vitro selection procedures to kind of isolate functional RNA, DNA, proteins from large pools of random sequences of these biopolymers. And at that time, I guess that's when you starting with RNA world kind of approach to origins of life. But then something that I really appreciate from Professor Szostak's in Professor Szostak's career is how he went out of his comfort zone and explored, made a kind of 20-year exploration of protocell, so connecting the RNA world approaches that he had been developing initially, but opening up to investigate also protocell, the protocell camp, which was for me a very an inspiration and I think we're going to be listening to maybe things that have to do with the two things. So what's the more or less? Or you?
Sure.
Some
of both.
Some of both. So, Jack, whenever you want, the floor is yours.
Okay.
I'm really looking forward to your message.
Okay, thank you, Kepa. Great, thank you very much. It's wonderful to come back to Donostia, San Sebastian. We always love visiting here and really appreciate the interest of people in science. It's really special to see that here. Okay, so what I'm going to talk about today is the origin of life on Earth when our planet was young. That what you see on the slide is just an image, a diagram of what we think the first, most primitive cells might have looked like. But before I go into more of the details, I just want to ask you, how many people think that life is everywhere in the universe? Lots of, yeah, there's some, right, out there on other planets around other stars. And how many people think that life is so special and complicated that it might only exist here? Some people, okay, so yeah. See this, I find this very interesting because of course we don't know, right? And yet people have often strong opinions that yes, life has to exist out there, or maybe it's so special it can't. So what we're trying to do in my lab and many of my colleagues is try to understand how life actually did get started, and from that, can we learn something about whether life might be easy to get to and therefore common, or it's very difficult to get there, in which case life might be rare. Okay, so there are actually very good reasons for thinking that there should be life, out there in the universe. One is there are so many environments that we know could support life, right? So we know now from the work of our astronomer colleagues that most stars have planets. And of those planets, there's a fraction that where they're at the right distance from their star, the temperature is like here, it can have liquid water. We know there could be life there, but we don't know if there is life. The other reason for thinking that life might be common is that even on our planet, there was so much time available for life to get started. So in between the time after the moon forming impact, the Earth cooled down so we could have liquid water. From that time until the first really clear signs of bacterial life on our planet, at least 700 million years. Somewhere in that 700 million years, life got started. We don't know when. We don't know if it popped up many times, or only once, in some special place, because there's no evidence left from that period of our planet's history. Okay, so those are two reasons for thinking that life might be common in the universe. But on the other hand, if you look at a diagram like this of the metabolism that's going on in every cell on Earth, you think, how could something so complicated possibly arise spontaneously all by itself? And actually, the problem is even worse than it looks like from this diagram of metabolism. Because if you look underneath that at the flow of information, of course, we all know information is stored in DNA, transcribed into RNA, translated into proteins. Proteins do most of the jobs in the cell. But the problem with this is that every part depends on every other part, right? You can't replicate DNA without help from RNA, without enzymes, without protein enzymes, and without the metabolites that are made by the protein enzymes. Same thing with RNA. You can't make RNA without the information coded in DNA, without the protein enzymes that transcribe DNA into RNA, without the metabolites that are made by protein enzymes. So, and so on and so forth. Every part depends on every other part. And so how could such a system emerge spontaneously? Right? And that puzzle was a huge paradox for decades. And there were many completely crazy theories for the origins of life early on. But actually, in the 1960s, there were a few smart people who said this molecule in the middle, RNA, could be the key, because when the structure of transfer RNA, the molecule that brings amino acids to the ribosome, when that was solved, you could see it had a complicated folded shape, kind of like protein enzymes have. And so people like Carl Loza, Francis Crick, Leslie Orgel suggested that maybe life began with simple cells with an RNA genome and RNA enzymes. And that theory is now known for short as the RNA world hypothesis. Nobody took those ideas seriously until the early 1980s, when Tom Cech and Sidney Altman discovered actual RNA enzymes encoded in our genomes and the genomes of other organisms. And so that immediately simplified the problem of the origin of life, because now we don't have to worry about getting everything all at once, right? DNA, RNA, proteins, metabolism. We can start just by thinking about RNA. RNA, we know, can code for information, can transmit information. We see this in RNA viruses even today. We also know RNA can act as an enzyme. And so maybe the idea was RNA could act as an enzyme to catalyze its own replication. So much simpler. DNA could come later, proteins could come later, complexity of metabolism could come later. And now, for the first time, we could imagine primitive, simple cells with only one biopolymer. Okay, so now we have a much easier problem, but still very, very difficult problem. We have to break it into simpler parts. Actually, first, let me just show you one picture for probably the best evidence, the strongest piece of evidence that life did start with this earlier RNA-based kind of cell. This is a view of part of the ribosome, the machine that makes all proteins in all cells. And in this image, the gray squiggles are RNA, the gold squiggles are proteins. And do you see where this little green structure is in the middle? That's where peptide bonds are made. And you can see there's only RNA near that. And so RNA is catalyzing the synthesis of every peptide bond, every protein that's made in every organism on the whole planet. The only logical deduction from that is that RNA came first and evolved the ability to make proteins. Okay, so if we can accept that, then we can start to look at some simpler problems. Break this big question down into some simpler questions. They're still very big and difficult, but at least it's a start. So we can, if we want to understand how RNA-based life first emerged, we can ask, how were the building blocks of RNA and other things, other parts of biology? How were they made on the early Earth? Once you have these chemicals, right, they're still not a living system. So how do a bunch of chemicals get together and start acting like a living cell? How does that happen? That's actually the part of the story that my lab has worked on most over the last 25 years. And finally, a question that we're just starting to be able to think about better and better is what kinds of environments on our early Earth would have nurtured the formation and the reproduction of the first cells. Okay, all right. So there are several popular theories for this first question, where did the building blocks of biology come from? And so I want to start by giving you 3 examples of where they did not come from, even though they're the most commonly talked about theories. And then I'll say a little bit about what our modern understanding is of that early chemistry. So you may have heard of the idea that life started in deep sea hydrothermal vents. And that theory is kind of popular because there's a source of energy deep in the ocean. It's shielded from ultraviolet radiation, and there's lots of life there now. When we go and look, we can see abundant life. However, this is an example of what I call pathological science. This theory has been talked about for 40, 50 years with absolutely no evidence. There's no evidence that you can do the chemistry that you need to make the molecules that we need to get to life. And so I think this theory is really something that we need to stop talking about it. It's just so wrong. Okay, there's another interesting theory, which is the building blocks of life might have come from asteroids, meteorites. And there's a lot of beautiful work on this over the last 60 years. And just in the last few years, we actually have sent missions to 2 asteroids. This is Bennu, which was visited by a spaceship which grabbed a sample of this material, brought it back to Earth, and people have been analyzing the chemistry, the chemicals that you see in that sample. And it's kind of ironic, in my view, that the scientists who did that work were very, very disappointed in one thing. And that is, the molecules of life are typically they have a handedness. They're either right-handed or left-handed, right? And for decades, people analyzing samples from meteorites that fell to the ground found an excess of the handedness of the molecules of biology, especially amino acids. And the idea was, by going to this getting a pristine sample from an asteroid, we could see for sure if that's the case. And everything they found in Bennu and also Ryugu is completely racemic, equal amounts of right and left-handed molecules. So that evidence, together with the fact that you see not just the right molecules, but thousands, millions of different compounds, tells me that this is not really relevant to getting the molecules to make life. Maybe a source of carbon or simple things like cyanide, but it's not where the building blocks of biology came from. Okay, and finally, there's the famous Miller-Urey experiment that was done at my current university in the year that I was born. And this was a real breakthrough experiment. So Stanley Miller was a young graduate student who's working in the laboratory of Nobel laureate Harold Urey. Stanley wanted to see if he made an artificial atmosphere mimicking what they thought the early Earth atmosphere was, give it a source of energy like electrical spark, what would happen? And his advisor, Yuri, said, that'll never work, don't do it. But Stanley did it anyway, and the result was spectacular, right? Because they found amino acids, which is one of the iconic sets of building blocks of biology. So it was a breakthrough in the sense that it showed people that at least some of the chemicals for life are really easy to make, right? The problem is that you don't just make those. You, again, like in the asteroid chemistry, you make millions of compounds. The harder you look, the more you see. And the right molecules, the molecules you need to build cells, are an incredibly tiny fraction. They're parts per million or usually parts per billion. So again, there's no way that you could start to construct more complicated structures if you have tiny amounts buried in a sea of other undesirable compounds. Okay. So what is the answer then? And I think over the last 20 years, this is becoming more and more clearly understood through the work of many people, but I think most importantly through the work of my colleague John Sutherland in the UK and many of his students and colleagues. So the basic idea has actually been around, which is that cyanide is the best starting material to make the molecules of life. And I really love that because it's a little bit ironic that cyanide, which is a terrible poison, is actually the best place to start with if you want to create life. Okay, but the reason is that cyanide has the right elements, has hydrogen, carbon, nitrogen, has a lot of energy stored in its structure, and it can be used to build up all the right kinds of molecules. But there's a problem. So cyanide can be made in the atmosphere of the Earth when it was young, not now, but in the early days, because the atmosphere would be exposed to high energy ultraviolet light, also lightning, impacts of large asteroids or planetesimals. That energy rips things apart to atoms, and a lot of them come back together in the form of cyanide. So that's all very good. The trouble is, when cyanide rains down to the surface of the Earth, it's in a very, very dilute solution, right? And so it would flow into rivers and lakes and the sea as a very dilute solution, and it wouldn't do anything interesting except react with water and hydrolyze and turn into useless products. So for a long time, that was a serious problem until Sutherland and his colleagues Dimitar Sasolov and John Gratzinger, who's a geochemist, realized that there's a simple solution to that problem. And that solution comes from environments that are hydrothermally active. So things like impact craters. You have a big object crashes to the surface of the Earth, makes a huge crater, it fractures the rocks, there's a lot of heat, and water will then start to circulate through those fractured rocks and bring material up to the surface. The same thing is true in volcanic areas. So, for example, Yellowstone National Park in the western US, it's volcanic, there's a lot of fractured rock, there's circulation of water down where it gets heated up, comes back up to the surface, and when that water comes up to the surface, it bears ions that have been extracted from the rocks. And there are many of those, but for this purpose, the important one is iron. So iron that's leached from the rocks comes up into the surface waters, and iron reacts instantly with cyanide to make a complex called ferrocyanide, which is very stable. And so it can accumulate over time, and there are some salts of this material that can precipitate out. And so together with sediments, you could build up, over thousands of years, a thick reservoir of this complex of cyanide. Okay? And so you can imagine these lakes. We can see this from the history of the Earth and Mars. Lakes sometimes dry up. If the lake dried up and then there was a lava flow that covered this cyanide reservoir, it would be heated up. And when you heat up these molecules, they're processed and they release cyanide, ammonia, other good building blocks like cyanamide and related compounds. But now, as a very, very concentrated mix. And so again, when it rains, the groundwater starts to percolate through this thermally processed material. It will travel again underground, eventually come up to the surface. And here's another picture from Yellowstone. Now you can imagine this concentrated stew of reactive building blocks flowing over the surface, being irradiated by ultraviolet light from the young sun. And remember, there's no oxygen in the early atmosphere because there was no photosynthesis to make the oxygen. So these compounds get, they do a series of reactions when they're exposed to ultraviolet light. And in a few simple steps, the cyanide is transformed into the smallest, simplest sugars and a series of other products. And so these simple sugars like this can react, make intermediates. So this small molecule goes on in several steps to build up the nucleotides that are the building blocks of RNA. And a lot of that pathway was worked out by John and his student Matt. And this particular one has amazing properties in that it tends to crystallize out. This is just a particularly beautiful large crystal of RAO, a photo taken in Donna Blackman's lab. So the idea is now, instead of having everything happen in a primordial soup, right? Now we take the right molecule, it crystallizes up, you get, again, a reservoir of this crystalline material. All the stuff you don't want can get washed away, and you're left with purified material that can then go on to do the next step in the process. And so here's this. So this has been a change in the kind of philosophy of how to do chemistry, going away from classical synthetic organic chemistry where you mix A with B and you want one product C and you purify that. Here, there's this new buzzword called systems chemistry where you take advantage of everything that's around. If sometimes you can make multiple useful products. Sometimes there are things like phosphate that aren't part of either the precursors or the product, but they catalyze the reaction. So it's a more integrated, holistic way of looking at the chemistry, which is, I think, is essential for understanding the chemistry of the early Earth. Now, this crystallization of RAO has another completely unexpected and amazing property that's highly relevant to the origin of life, which is that in a magnetic field, it can crystallize out on a magnetized substrate, and only one mirror image form of the molecule will crystallize out. And so in that one step, you can, again, remove the wrong material, wash it away, and the right mirror image molecule that gives us what we need in biology can be purified in this way. This is amazing work from Szostak and Sasolov. Okay. Finally, let me just show you one more of these critical chemical reactions that relies on UV. So here is the intermediate after RAO. You see the blue part is the nucleobase. It's the information specifying part of a nucleotide, but you can see it's below the green part, which is the sugar, the ribose. Okay? This doesn't work to make RNA. You need the nucleobase to be above the sugar. This reaction was known for 50 years, but no one could get it to work until they realized that if you have a sulfur here, exposure to ultraviolet light will flip the nucleobase to the top, and then presto, there you have. You have the molecule that you need to build biologically relevant RNA. I mentioned phosphate briefly. Phosphate is part of the backbone of RNA and DNA. Right? So we know that it had to be there, right? But for 50 years, there was the so-called phosphate problem, right? Phosphate reacts with calcium. It precipitates as a mineral that's highly insoluble. And so nobody could figure out how could you ever get enough phosphate in solution to actually make the molecules of biology, like RNA. Until David Catling, a few years ago, realized, well, actually, there are a few lakes on Earth today that have very high levels of phosphate. And why is that? And it turns out they are known as the alkaline carbonate lakes, like this lake in British Columbia. The phosphate can come in, it's leached out of rocks, very dilute, but it can accumulate as long as there's no biology there to absorb the phosphate and reduce its concentration. So in this particular place, phosphate builds up to high levels, something that could clearly have happened on the early Earth. Okay. All right, so I've talked a little bit now at just sort of a high level of how to make the right molecules, but then what happens, right? After all, we don't want just a bunch of chemicals in solution. We need to understand how they come together to make structures like cells. So this is a diagram of what we think the important components are. There are two of them. One is a cell membrane that provides some spatial localization so that everything, all the contents don't just float around randomly. And inside, some small pieces of genetic material that we think are probably RNA or something very close to RNA. Okay? So it turns out making structures like this is very easy. It will happen all by itself. If you have some soap molecules, they'll assemble into membranes, they'll capture whatever's around when they close up into a spherical shape, a vesicle. If there's RNA floating around, some of it will end up on the inside. That part's easy. The hard part is how does something like this grow? How does the membrane grow larger? How do we feed it with new material so it can grow? And how does the RNA get replicated before there are any enzymes? Right? These are huge puzzles. And how does division happen before there is all the complicated machinery that modern cells use to control when and how they divide into daughter cells? Okay, so I want to talk a little bit, just briefly, about first the membrane part of these simple cells, protocells, and then show you just a little tiny bit of some of the recent work that we've been doing in my lab on the RNA side of things. So I really love these primitive membranes that are made from fatty acids. Okay? So this is basically just soap and water. There's nothing more complicated than that. This fatty acid is what comes from olive oil. These are shorter. This one we think is more likely to have been made on the early Earth. Any of these, you shake them up in water, they'll make these beautiful structures like you see here. So you can see a big vesicle and smaller vesicles trapped on the inside. One thing that I find really fascinating about these structures is that what they form depends very sensitively on the environment. So if the environment is too basic, too alkaline, they make tiny, tiny structures called micelles, just a few nanometers in size. As you slowly drop the pH more towards neutral, they'll all of a sudden start assembling into bilayer membranes that look kind of like modern cell membranes, but they have some very different properties. If you drop the pH too far, it becomes acidic, then these collapse and they form oil droplets. So if you're in the right environment, the right conditions, or the right acidity and salt and so on, they'll form structures spontaneously that look just like what you'd need to build a protocell. let me show you another property of these vesicles that I really like. The membranes made of fatty acids are very dynamic. The molecules that make them come in and out of the membrane and go from one vesicle to another every second or so. Very rapid exchange. But the vesicles themselves maintain their identity indefinitely for weeks or months. So in this experiment, we made one population of vesicles labeled with a green dye, another labeled with a red dye. We mixed them together. You can see this after a day in this photo, they're still separate, right? They've maintained their own individual identity, but they're made of different molecules every second. Their component molecules are different. So it's kind of like us. You know, our molecules are different over a period of years. Some say 7 years. We're made of different molecules because we bring in food and you know, to excrete wastes. So it's a similar thing, but on a faster time scale. This dynamic property turns out to be really important for how these things can grow and divide. So what we can do is just add more of these fatty acids. Some of that material will get into the membrane, which will then grow, and then division can happen spontaneously. So I'm just going to show you a movie of an experiment that was done by Anna Wang when she was in the lab. So here you see vesicles. The green is RNA labeled with a green dye. You might see an orange dye. We've added more fatty acids. Now the surface area grows faster than the volume. The surfaces can start to fluctuate. You get all kinds of crazy shapes. Every now and then they pinch off, release daughter vesicles. And so in a very simple way, you can feed them, watch them grow, and watch them divide, just in a one simple experiment. Okay. Okay. There's another. There's a fascinating phenomenon, and so there are many people who actually are very skeptical that the first form of life could have used a membrane envelope, because you have to get food across the membrane to the inside in order for the RNA molecules to replicate. And how could that happen? If you think about modern membranes, our cell membranes, it could not happen. No way. Our membranes are good barriers, and everything gets across. Everything that gets across in or out is controlled by proteins. But these membranes have a very different property. Molecules can come from the outside, approach the surface, make complexes with head groups of the fatty acids, and just sort of invert across the membrane. And so even big charged molecules can cross these membranes spontaneously. So very different property from modern membranes, but exactly what you need for a primitive membrane. Okay. Let me then just very briefly say a little bit about this RNA, the primitive genetic material. Right here, the big puzzle is how could this material replicate? Right? How can you copy an existing strand, make a complementary strand, then copy the copies? This has been a puzzle that people have worked on for more than 70 years now. A lot of the early work was done by the late Leslie Orgel, I mentioned before. He's a real pioneer in this field. And he was the first one to study the polymerization of nucleotides that are chemically different. They're much more reactive than the nucleotides in our bodies. Okay? And we started studying this maybe 10 years ago. And I'll just show you a surprising thing that we found. So in biology, if you have a strand like this black strand that is growing by the addition of monomers in a way that you can copy a template strand by making Watson-Crick-Franklin base pairs, in biology, one of these comes in at a time, binds to the template, reacts with the primer, grows by one nucleotide, and you repeat that process, perhaps millions of times. So we were pretty sure that this early prebiotic chemistry should work the same way. And it took us years to figure out that actually it didn't work that way. It's completely different. What actually happens is that molecules like this react with each other to make this more complicated intermediate, that you couldn't dream up structures like this, right? Nobody ever thought of anything like this. And it turns out these form in solution. These combine to the template by making 2 base pairs. So they bind more tightly, which means you need less of the material, lower concentration. And this structure here in 3 dimensions is organized perfectly for the attack of the primer hydroxyl on the adjacent phosphate. So it's predisposed to do the right chemistry. And as a result, these form, they bind, they react with the primer, it grows by 1, and then you release 1, which can then go on and repeat the process. So that's how we think primitive copying worked without enzymes. Okay? So we've made a lot of advances on that front, but now there's a bigger problem, right? It's all very well to copy a template strand, but if you make a long duplex, it's kind of a dead end because it's very hard to get long RNA strands apart from each other. And even if you can get them apart, they come back together again thousands of times faster than the copying chemistry. Okay? So it looked like a complete roadblock. And there are some other issues that I won't go into unless you want to ask questions later. So we were thinking about this during a period during the COVID pandemic. We couldn't go into our lab. We were stuck at home, nothing to do but think. So we became theorists for a little while. And we came up with this crazy theory shown here. And this is a model that I think solves all of the problems with going from copying to replication. Okay. So the idea, briefly, is that the genome of a protocell would be a circular sequence shown by this green circle. But there are no molecules that look like that. There are only fragments whose sequence maps to this consensus. And these fragments come from both strands. So when the conditions for base pairing are favorable, they come together. Sometimes you can do a little bit of primer extension, a little template copying, at places all around the circle. Right? Then, when that's done, you have some higher temperature or some other fluctuation, the strands come apart, and then later on they can base pair again. You can do a little bit more copying. And so the copying is done in bits and pieces all the way around this circular sequence. And again, biology doesn't work this way, ever. Right? In biology, you start at the beginning, you copy, you go all the way around, and you stop at the end. Right? But that's easy to do if you have fantastic, highly evolved enzymes. It wouldn't work if you're relying on simple chemistry and physics. So this is a way of doing copying and replication a bit at a time in a distributed fashion that can ultimately lead to replication. And interestingly, in a way that depends on fluctuations in the environment. Not a steady state environment, but an environment where temperatures go up and down, maybe salt concentration goes up and down, or the acidity goes up and down. So you can have these cycles where strands come together, you do a little copying, they come apart, then they come back together, and you do a little more copying in different places. If you repeat that process, you can do replication in a very simple way. Okay. The last important point I mentioned is where on our young Earth could any of this have happened? And I mentioned volcanic regions as one likely source. And so again, we return to Yellowstone Park. This is Yellowstone Lake. It looks very peaceful, but actually underneath, you have these vents which are releasing streams of hot water. And that led us to propose a model like this. We still think something like this is going on, maybe not exactly like this, but the idea is protocells could live out in the cold lake water. Every now and then, they get swept up in one of these thin streams of hot water that helps the strands come apart, nutrients rush in at higher temperature, and then they get swept back out into the cold water. And that's a good thing because you don't want to cook RNA. It is a delicate molecule, right? So a short exposure to high temperature is fine, but you wouldn't want to be hot for hours, for example. So again, it's a cycle driven by the environment. And I think this is one of the key points about primitive life. It hasn't evolved the internal machinery to learn to cope with different environments. And it hasn't evolved the ability to make its own nutrients. It's relying totally on the environment to supply everything it needs and also to supply the sources of energy and the fluctuations that could drive the primitive cell cycle. And so there's a lot of work going on to try to understand in more detail the kinds of environments that would have given rise to primitive life. So, just to sum up, what have we learned? We've learned a lot of facts about the origin of life. We have a lot of good models that explain a lot of puzzles. But to me, the most important thing that we've learned is that we have a lot of deeply ingrained biases in our thinking, a lot of preconceptions, sometimes based on how biology works, sometimes based on classical chemistry. And in order to make progress, you have to be able to let go of these preconceptions and follow the evidence to the truth, okay? And that's been incredibly successful in this field. And we've discovered so many crazy phenomenon that you couldn't just dream up by armchair theorizing, right? You have to do experiments and interpret them, and you can learn about how things really work through this combination of theory and experiment. Okay, and finally, I think we're actually at a point now where these new discoveries are bringing us very close to understanding how life might really have started on the early Earth. And I hope in the next few years we'll have a more and more complete understanding. Okay, so I've tried to mention some of the key people as I went along, but all this work was done, not by me, but by many, many fantastic graduate students and postdocs in the lab over a long period of time. The people in the bottom row are all in the lab now, and they're doing these experiments. I'm talking here, and they're back home working hard, I hope. And these are a lot of people who are recently in the lab, many of whom now have faculty positions at universities around the world. And let me just end by saying, if you want to learn more about this, there are many good books. There are books by my friend Carlos Briones. I wrote a book recently in collaboration with Mario Livio, who's an astronomer. My part of the book covers how life might have started on the Earth, and Mario writes about life out there in the universe, around other planets, other stars, and so on. And so I'll stop there, and thanks for listening. Thanks for inviting me back. I'm happy to answer questions.
Okay. So thank you very much. You deserve at least some water. So the idea is to establish a dialogue with the public, but I'm going to take advantage of my position here and just ask a couple of kind of general questions. And then we open the floor to the public, if that's okay. Is that what we are expected to do, more or less?
Okay. All right, here's where I'm in trouble.
No, come on. So there's a kind of a tension, and you explained it very well. Like, everything depends upon, like, the components of life. They depend upon each other, and you drew the central dogma there in a way that I like, with the loops in which, because sometimes they forget about the loops with the proteins and all the rest of it. But so this is a very convoluted system, and we know that the interdependencies are incredible. So in the late 20th century, when we had these discoveries that you spoke about ribozymes and the structure of the ribosome, and we realized that RNA had to be there, or at least oligonucleotides of RNA initially.
Short bits.
Short bits, maybe initially. So but it's kind of tempting to go to RNA molecules when they are, I mean, and there's a kind of a jump between small molecules like oligos, oligopeptides, because there is a kind of, when you are not big enough, the type of functions that you can perform are much more limited, right?
Yes, for sure.
But a way of simplifying the problem is without reducing diversity, because if you go to an RNA world idea, you are reducing kind of a lot of diversity, and that has an advantage, as you said, because you can concentrate on a few issues.
Right.
But don't you think that a better approach in terms of in Origins of Life, we have to simplify, because otherwise
Right, but we shouldn't neglect the problems that you've raised. And so I could say we're actually working on all of those things. In the model for replication that I showed, we have only small pieces of RNA, right? And as you said, they couldn't do much, right? But we want them to make larger molecules that could catalyze useful reactions. So how could that happen? That's a big thing we're working on. And our idea is these small pieces can assemble with each other. There's some beautiful reactions called like loop closing, like two oligos come together and then the end overhang can curl around and ligate and close up. You make a stem loop. You can do simple things like that and build up large, complicated RNAs that could act like an enzyme. They never need to get replicated. Right? So only the small pieces replicate. Okay. The big complexes do the functions. And so that's one way of resolving that kind of tension. We also have some chemistry that brings in amino acids. We're still trying to understand what the heck can peptides do. They must be doing something, but we don't know what.
Yeah, because in reality, we don't have self-replicating molecules as such, as you were also saying. Like, it's always in that the processing of that, you know, the DNA, RNA. So it's all in the end, the active molecules in the cells are proteins, right? The thing is that we need to create proteins, you need the DNA, and that's the chicken egg problem that you were referring to. And but in reality, there are not self-replicating. So if you look at what happens in the cell, there are template. So there are processes where template helps in the replication. But it's exactly
right. So there are many people like my colleague Jerry Joyce, who are trying to evolve an RNA enzyme that's a good enough RNA polymerase to copy its own sequence. He's been working at it for, 30 years, getting closer, not there yet. I think our model will get to replication sooner.
One day.
We'll see.
And we will learn from it, I'm sure,
But how to get to proteins, right? This is a huge problem. And actually, John Sutherland, whom I mentioned, John and I have arguments all the time about the origin of protein synthesis. He thinks it came in very early in a simple way. I think it's so complicated, it must have been many steps, a long process of evolution to get to the ability to make coded proteins. His former student, Matt Towner, who is a postdoc, has recently published a way to make peptides that are not coded. So then we argue about, well, what good is that? Can they still do something useful? Yeah, there are a lot of big questions like this.
So what does John think about the work of Towner? Because I've heard different stories about this. Anyway, maybe we get into trouble there.
It's beautiful chemistry, but then the question is it relevant to the progression towards ribosomal protein synthesis?
Okay.
It's
an open question. Okay. Then one more, and then I'm a bit. But the idea of metabolism that you spoke about, right, and how so when do you think because the synthesis of all of this, as you say, has to be we need energy resources. They are far from equilibrium systems, and So when do you think the metabolism, even simplified versions of that, like what Ram Krishnamurthy could be working on these days, on simple versions of the citric acid cycle, like what do you think about models of origins of metabolism that don't need to go all the way to that complexity that you drew initially, but somehow drive also the system and couple with
Right. The thing about evolution is every small step has to provide an advantage, right? And that's a really important clue. So if we want to think about the origins of metabolism, we have to think about it in a stepwise manner where catalyzing one reaction provides an advantage, and then maybe catalyzing a different advantage or the previous reaction, you know, and you build up pathways. This is an old idea, but I think it's still valid. The question is what reactions would be, say, the first to evolve, that would provide an advantage for the cell. And we have almost no clue about this. I think one of the things I hope is if we're able to make protocells that can start to evolve in the laboratory, maybe we can see experimentally, right, what useful reactions would evolve first. I think in the next 10, 20 years, this is going to be an area where the biggest advances are made. Origin of metabolism, origin of protein synthesis, origin of membrane transport machinery, all of these things kind of related. So yeah, the doors are just opening to that now.
Thank you. Thank you very much. Now that my curiosity was satisfied to that extent, to a certain extent at least, let's open the floor to the public and how are we gonna? So there was a hand there. Yeah, can we get the micro there? Yeah, there was a hand very early there, and then there is another one here. Yeah.
Given that there were only a few 100 million years that there were habitable conditions on Mars, would you expect, to which stage would you expect to find life forms on Mars? RNA world, or what would it be?
Yeah, I think this is one of the most exciting things is our exploration of Mars. Just, you know, first of all, to understand the geology, the ancient history, because on Mars we actually have evidence. We have landforms that survived for 4 billion years, right? It's all gone on the Earth. So we know where to look for early signs of life. And we also know where to look, you know, there's a possibility there might be life on Mars, but it will be in salty brines that are underground. So we have to have the technology to go there and drill underground, get samples, either have advanced labs on Mars or bring them back to Earth. So I would not be surprised to find life on Mars in those, you know, sort of refuge environments. But my bet would be that it would share a common ancestry with life on Earth because there's been so much exchange of material between these inner planets over time. But we'll see. Yeah. I would love to be wrong. We'll see.
Thank you.
There and then there. Yeah. To the center.
Perdónenme la pregunta, pero es que me parece no haber entendido nada de qué es lo que al principio se nos dice. ¿Cómo? Formas en las que no surgió la vida y nos aparecen después todas aquellas que usted pone en una diapositiva y va detallando ya con sistematización cómo pudieron darse los pasos que dieron lugar a los enganches de las moléculas distintas, a la deposición de los fosfatos, pero todas ellas aparecen en la diapositiva que nos puso al principio de cómo nos originó la vida. Y entonces yo no me aclaro de nada.
Okay, he's a bit confused about the fact that in your slides, you look like you're solving the problem where you said that it was not solved initially, and the things that you... So I think, well, I don't know, you can address it or I can address it as well. Like,
yeah, well,
so
there are some, it's a huge puzzle, right? With many, many steps, many pieces of the puzzle that have to fit together. Some little parts we understand pretty well. Many parts we don't. I mostly presented the few things we understand and not leaving out a lot of the parts that we don't understand. But hopefully, progress is being made. I think eventually we'll see how all the pieces fit together, and we'll have understand the whole pathway.
Yeah, thank you for the wonderful explanations and lecture. I've got a question. Is it possible that the laws of the very small, as established by quantum physics, could they have an impact on the structures and processes that you describe?
Absolutely, absolutely. The best example actually is this separation of the mirror image isomers of this molecule RAO on a magnetized substrate. And this goes through a quantum phenomenon called CIS, the chiral-induced spin selectivity, that it goes very deep into quantum mechanics, far above my head. I don't really understand it, but it's fundamentally quantum mechanical phenomenon that leads to a macroscopic effect on crystallization. And there are probably many other examples. And I mean, at some level, all chemistry is based on quantum interactions, but this is I think, a good recent example. Yeah.
please.
Thank you very much for the amazing talk. I have two questions. In your picture, what's the role of surface catalysis in the origin of life, since what you have described is mostly looks like is in solution?
Yeah, yeah.
And second is the idea of, as Kepa mentioned, metabolism on those surfaces, because as far as I know, it has been described some primitive, non-driven by enzyme metabolism in rocks, for example, primitive glycolysis and that kind of thing. And finally, related to that, what's the role of the scaffold on RNA? I mean the sugars chain.
Okay, so let's see. Start with surface catalysis.
3 in the end, yeah.
Yeah. So
for the same price, no?
Yeah. So there was some beautiful early work by Jim Ferris and together with Leslie Orgel on the surface catalysis of assembly of RNA from activated building blocks, and also surface catalyzed assembly of long peptide chains, especially from acidic amino acids. Question is, how can that be relevant when you have protocells, right? Maybe there's a way, but I can't really see how you could have mineral catalysis inside protocells where the minerals also have to replicate and be distributed to daughter cells. So maybe it has a role in setting the stage, but once you have protocells, I think then everything is probably in solution. Or, actually something we've thought a lot about is surface catalysis on the membrane surface, which is a very interesting possibility. We've studied that a lot. Nothing we've tried has really worked, so it's a little disappointing, but it's a possibility. And then what was the other?
The sugar bone.
Oh, the backbone, yes. So the backbone of RNA is the sugar phosphate. And it turns out you can make molecules that work like RNA using different sugars. The most famous one is where the sugar is a 4-carbon sugar instead of 5 carbons. This was TNA, invented by Albert Eschenmoser. It base pairs with itself, it can base pair with RNA, it can base pair with DNA. It looks like it could work just as well, but when we tried to do its copying and replication without enzymes, it's completely dead. So it's very interesting. We don't fully understand, well, I think we do understand why it doesn't work. It's because of the stereochemistry of the backbone. But yeah, there are other things that can work. I think everything we know so far points to ribose as being the easiest way to get to RNA, partly because of, for example, crystallization of RAO, which is a critical step on the pathway. So it may be, out of all the possibilities, RNA is the easiest one to get to.
And the third question was more kind of related to whether you can have a two-dimensional kind of metabolism. Was that the like the Westerhuizer type of or whether was that?
Yeah. Okay. So there's this interesting chemistry with iron sulfide-based rocks, right, minerals, and in biology you use iron sulfur clusters. So they have the same shape and the same chemistry as these rocks. But you don't need to use rocks. You can use soluble small clusters to do the metabolic reactions. I think the rock stuff was kind of a red herring, a distraction. Sorry?
Please, Álvaro. Álvaro is my mentor, philosopher of biology. Álvaro Moreno, do you have the micro there for him? Yeah, it's coming.
Well, first of all, nice talk. Congratulations. Thank you. But my question is the following one. Why is it, in your view, that all of the steps that happens during the path through our life, all the new steps seem that have erased the previous one, and yet once life, prokaryotic life as we know it, all the following steps in the direction of complexification have not erased this form. On the contrary, depend on this form. So it seems that there is a kind of deep turning point or something that is completely different. So it seems that there is a process, and a given moment, all the previous steps to this process erase the previous stages, and yet, once this step is achieved, then all the 4 billion years later of the evolution kept this form and depend on this for the further steps.
Yeah, that's a very deep question. And not surprisingly, there are a lot of arguments about it. My friend and colleague Nick Hudd at Georgia Tech.
Yes, Georgia Tech.
He actually thinks that life started from something before RNA, kind of a scaffold that then was the start of biology, and then biology somehow evolved the ability to code information in RNA and then got rid of this earlier step. Okay, I find that a little bit hard to accept because this kind of genetic takeover, once you're doing multiple things with some genetic material, I think to change it all at once is very hard. So I don't know. I mean, I could be wrong. Maybe his idea will work out in the end. But I think it's actually easier to imagine or to see that once you're dependent on something like RNA, to change it to something else would affect everything. And so making a change like that would be almost impossible to do. So I think the whole idea of genetic takeovers is very, very difficult. And that's why life still relies on this earliest RNA molecule. It's kind of trapped into using it forever, because it would be too hard to change.
So in a way, your answer is that not everything has been erased. No, that there is a kind of a there is a kind of a line that remains there and across the stages.
Yes.
But the actual fact that there is a gap, a big gap between what we can implement physico-chemically, even if it's complex mixtures of compounds, and what life does. The gap is amazing, isn't it? Like,
I think the gap is being narrowed bit by bit.
Okay.
There's a brilliant chemist who is a student of Matt Powner, who recently came up with a way to make one of the most complicated cofactors, acetyl-CoA, by prebiotic chemistry. Simple, non-biological reactions. So I think biology today has many relics or fossils of our origins.
Any other question or comment that you?
Quería preguntar sobre la particularidad de la Tierra que no tienen otros planetas, que es la existencia de la Luna. Y me refiero a que, según se supone, en un comienzo, cuando estaba la Luna mucho más cerca que de la Tierra, al comienzo de la historia de la Tierra, esto generaban unos efectos de marea que eran de kilómetros de altitud. Si había unas mareas continuas de tanta altura, de kilómetros, es lógico suponer que en los puntos calientes de la superficie, en los que estuvieran, por supuesto, submarinos, el agua llegara a alcanzar su punto crítico por la presión enorme, y después bajara esa presión y desapareciera. Y esto fuese una cuestión cíclica, pasar al punto crítico y dejar de estar en él. Estando en el punto crítico, el agua es un disolvente universal que disuelve también los alquitranes, todos los que se forman con los experimentos de Miller y similares. ¿Esto pudo tener alguna influencia que no se encuentre en el exterior?
Okay, many things there, but he started the question on about the influence of the moon. So the idea that we had the Earth had a moon, and how the tides were huge, and that, and then he connected that with the properties of the water that could be changed as from the influence of the moon. What do you think about this? And also kind of, what do you think about it? I made it short, sorry, but...
That's related to the question of, on the early Earth, how much land surface was there? How high did the land go above the ocean? How deep were the oceans? How much of the surface of the Earth was covered with water? And these are all big unanswered questions. I think most people would accept that at the least there were volcanic islands like Iceland, Hawaii, Japan, and those might have been the environments, and if they're high enough, right, and not washed away by the severe tides, that seems like a good environment, but that's the severe early tides. That's a very interesting point. It's a good puzzle. It might have made it hard for there to be much land surface early on. It's a good point.
Yes.
Hi, good evening. Thank you very much for a very interesting presentation from someone who is really from the letter side of studies. I was wondering about the application of these, what I suppose is fundamental scientific research. Is there anything applied in environmental sciences or in medical sciences? In the same way that we're talking about the origin of life, I'm thinking about the death of cells, so death per se, and how to fight death. I'm wondering.
So my usual answer to questions about applications is I don't think there'll be any. It's just, we're just doing this because we're curious about how we got there. But I have to add that when you're doing really fundamental science, basic work, it always surprises, right? Things come up where there are applications where you least expect them. And some of the chemistry that people in my lab developed or worked on has led to some applications that led to an unexpected collaboration. So they provide some useful tools. I would say the fact that I'm probably one of a small group of scientists who have worked on both RNA and membranes led me to get involved with a biotech company called Moderna, which was one of the two companies that made mRNA vaccines. And a lot of what we talked about at Moderna was, how do you make nanoparticles with lipids and RNA and get them into cells? So not a direct application of our studies on the origin of life, but the technology and the ideas are related. And so there's some cross-stimulation there.
Anything else? More comments? Come on, don't be shy. Please.
What is the time scale on which molecular complex crosses in the membrane?
Oh, yeah, I wish I knew. I think...
Okay.
Yeah. Okay.
That's okay. Just keep...
To repeat?
Yes. Please, can you repeat the question on the microphone so that everybody...
On what time scale does a molecular complex cross a membrane?
Yeah. So the short answer is we don't know. But so we can get the molecules we want across these fatty acid membranes in minutes to an hour. But that's different from how long it takes one molecule to flip from the outside to the inside. That could be a microsecond or less, but to do it many, many times and get enough material to the inside of a protocell takes minutes to hours.
So maybe one could attempt computer simulations to clarify this.
Oh, we do, yes. We use computer simulations to investigate it, indeed, we do.
And to complement the
experiments.
And also to complement your answer, it will depend also both on the properties, of course, of the and of the compound. If it is charged, if it's not charged, if there are defects. There's a beautiful paper by Sheriff Mansi in 2010 in which he actually addresses this, how small defects in the bilayer can also help. So it depends on the properties of the membrane and the compound that you're. So it's even worse in the sense that it's more complicated. Yeah. All right. I'm gonna. So there was a kind of a suggestion of a connection with aging in the previous question, and this kind of makes a circle with your original, your youth. Let's say there's increasing evidence that even prokaryotic life. In prokaryotic life, there are pathways for programmed cell death. To what extent do you think that that happens within, I know that you're focusing like me, getting the thing started, right? Yes, it's hard enough.
It's hard enough.
It's hard enough. But then we see that even cells that are very, relatively simple compared to, so for eukaryotic cells that are the cells that make our organisms, we actually need for tissue homeostasis and all that, we need that some cells die because otherwise, and they actually commit suicide for the benefit of the rest. And that's the way we keep all the balance between our tissues, our organs, and all of that. Eukaryotic cells, of course, have different ways of, is the intrinsic pathway, the extrinsic pathway, but there's increasing evidence that even prokaryotic life, connected to the intrinsic the intrinsic pathway, but it's not. So, when do you think the aging and death or these kind of things, programmed cell death came about? And maybe with that we can start closing the circle here.
I think for programmed cell death, that must be the result of a long path of evolution. Because that usually occurs when cells are trying to build a more complicated structure that has many cells, and some cells have to die in order to build the whole structure, right? So that has to be a highly evolved process. But some of the fundamental things that lead to aging are not just telomeres and the ends of chromosomes, but many other kinds of damage. Damage caused by ultraviolet light, damage caused by protein aggregation, damage caused by oxygen or reactive oxygen species. Many things that contribute to aging, and that's why it's both so complicated and so interesting and so hard of a problem to solve.
Okay, so let me just finish then with another applause. Thank you very much for coming. I think that Donostia is so... You want to say something? For tomorrow. Okay, but tomorrow we'll have another session. It will be a round table, and you are all welcome, and come with more questions, and we will try to address them, and it's wonderful. So thanks very much, everybody.
Thank you. Okay.